Good day, everyone. My name is Rodolin Mercado Busa, and I'm here to discuss a research article concerning Ecology and Evolution of Melanism in Big Cats, case study with black leopards and jaguars by Lucas Goncalves da Silva. So let's get started. Next slide, please. Let's first talk about the abstract of this study. There is such a variation in animal coloration that have intrigued the evolutionary biologists for a very long time. Among the observed pigmentation, polymorphism or melanism has been reported in multiple organisms, influencing several biological factors, and classical hypothesis has suggested that such variant can present in adaptive advantages under the certain ecological conditions. In leopard, or as Panthera pardus, and jaguars as Panthera onca, melanism is caused by recessive and dominant mutation in the ASIP and MCIR genes respectively. So these chapters focus on melanism in these two species, aiming to analyze its geographic pattern. The frequency of melanism was 10% for both species. In leopards, melanism was present in 5 subspecies and strongly associated with moist forests, especially in the Southeast Asia. In jaguars, melanism was totally absent from open and periodically flooded landscape. In contrast, forests displayed a frequency that has similar to the expectations. So the analysis of the environmental predictors suggests a relevant role for the factors such as in the moisture and temperature. These observations support the hypothesis that melanism in big cats is not a neutral polymorphism or influenced by natural selections, leading to a non-random geographic distribution of this coloration phenotype. Since we already elaborated the research objective of this study, let's now move on and explore deeper. So, the adaptive relevance of animal coloration has been explored and discussed for over a century. So let's talk about melanism. What does it mean? So melanism is the color polymorphism that is common in the various groups of organisms in which the skin, the fur, or the plumage is darker than what would be considered as normal or wild phenotype. Melanism can provide an adaptive advantage in certain ecological conditions, and exists the hypothesis of an association between the darker individuals and weather areas with dense vegetations, such in the tropical forest. Alright, so there are also some biological factors such as thermoregulation, susceptibility or response to disease, camouflage, aposematism, sexual selection, and reproductive success that could be directly influenced by melanism, and the occurrence of this melanism is rather common in Felidae. Next, how do we define jaguars and leopards? So, the jaguars as the Panthera onca. So jaguars are the world's third biggest cats and also the largest cat among the South America's big cats. So the usual color of their fur is tan or orange with the black spots. That was called rosettes because these spots are shaped like roses. Some jaguars are so dark they don't seem to have spots. Jaguars are considered as one of the big cats because of their size, strength, and predatory skills. Jaguars often live close to lakes, rivers, and wetlands that preferring to avoid open forests or grasslands. Next is for the leopard. So, and for the leopard, this is the largest spotted cat in Africa and Asia, and an important extant representative of genus Panthera. Leopards are big cat that was known for their graceful yet ferocious hunting methods and their golden spotted bodies. Leopards are bigger than a house cat, but considered as the smallest members of the large cat category. The leopard is very adaptable and can survive in many different places across the globe. So these large cats can live in almost at any type of habitat, including the rainforest, the deserts, the woodlands, grasslands, savannas, forests, mountain habitats, coastal crops, scrublands, and swampy areas. So, For our null hypothesis, or the HO, is melanism present throughout the species distribution that occurs randomly across all environments. So this hypothesis suggests that there is an absence of association between different landscape conformations and melanism. It is saying that the environment or the samples are living in does not have an effect on the genes of the sample which causes the melanism in them. And for the alternative hypothesis, or the HA, is melanism is distributed according to the biogeographic constraint. So yung biogeography is the branch of biology that deals with the geographical distribution of plants and animals. So this theory suggests that melanism does not randomly occur and that melanism in leopards and jaguars is not neutral polymorphism. So the figure 1 shows the location methods, the location record points from the entire historical range of the leopards that encompassing the various biomes such as the moist and the dry forest, grassland, and desert areas. And for the figure 2, it shows the location records at points from the entire historical range of the jaguars that encompassing the various biomes such as moist, dry forest, grassland, and desert areas. So these records were obtained from 5 different sources. The first one is individuals kept in scientific collection that possess information on the geographic coordinates of the sampling location as well as on code color. The second one is individuals captured or found dead during the field ecology studies. The third one is field collected fecal samples whose melanism status could be confidently interfered using molecular assay on or an investigate analytic procedure in laboratory medicine, pharmacology, environmental biology, and molecular biology for qualitatively assessing or quantitatively measuring the presence or the amount or the functional activity of a target entity. The fourth one is the camera trap records. And the last one is the samples available in online databases with the precise geographic origin and available sources information. So, the location records of individuals confirmed to be melanistic or non-melanistic were used in the statistical analysis based on the frequency of these phenotypes for both species. So, the researchers used the accumulated records and converted it into the degree coordinates using the WGS, or the World Geodetic System. This was the reference of the coordinate system and used to analyze information about natural landscapes in which the phenotypes occurs. For modeling the potential distribution of melanism, in the initial analysis, they considered 37 explanatory environmental predictors and landscapes data that covered 100% of leopards and jaguar that was known for distribution of International Union for Conservation of Nature, or the IUCN data. And they used of 35 environmental variables that was obtained from the WorldClim and Climon databases. So since correlation among explanatory predictors can lead for modeling of overfitting, they computed Pearson's correlation, or the coefficient between each pair of variables. So the correlation was assessed by extracting predictor information from 10,000 unique and randomly generated points within the present geographic distribution for the layer of both species. The statistical analysis of record distribution for each biome was performed using the chi-square test with Fisher correction, and the basic approach was to test differences between the observed and expected frequencies of melanism for each different ecoregion. So now let's talk about the results. So in leopards, there was about 623 leopard samples that was comprising the 556 non-melanistic and 67 melanistic individuals. Melanism presented a global frequency of 10.75% across the species range, with regional frequency varying among different landscapes, such as in the biomes and ecoregions. So the confirmed presence of melanistic leopard was recorded only in 5 of the 9 valid subspecies, such as in Africa, in Central India, in Bhutan, Sri Lanka, Southeast Asia, and Java. All of these regions contained new records for areas in which melanism had been previously described, as well as the representation of the additional areas. So melanism was absent in the leopard subspecies occurring in the Russian Far East, in the central China, in the Arabian Peninsula, and in the Middle East. So in this figure, it shows the location records of the database of the leopards that was divided by each valid subspecies. The differences among the biomes were significant, especially in tropical and subtropical moist broadleaf forest, where 30% of the animals were black, which is almost the time there were which is almost 3 times more than expected if melanism were an evenly distributed neutral polymorphism. In contrast, there was about 980 sample jaguars, 884 of which were non-melanistic individuals and 96 were melanistic animals were obtained. So the overall frequency of melanism was 9.80% across the species range. Interestingly, almost the same frequency expected for leopards. So most of the records of melanistic animals, which is the 92 in total, were located in South America. So the figure 4 was the potential distribution maps of the two coloration phenotypes that was analyzed in the study. The first one is the distribution of non-melanistic leopards, and the second one is the distribution of the melanistic leopards. So moreover, all regions that had previously been reported as potential sites of melanistic jaguar occurrence in different biomes of Brazil were corroborated with this study, especially with the Amazon and the Cerrado areas. Additionally, new records were obtained for areas where there were no previous reports of melanistic animals. When the presence of animals across the regions was assessed, they observed the marked differences in its frequency among distinct biomes and ecoregions. So the non-melanistic model provided a good fit to know the present broad distribution model for the jaguars in the Americas, indicating that it is provide a suitable baseline against which to compare the melanism model, as you can see on the figure 5. So the melanism model showed some similarities with common model, but also some important differences, especially the low suitable habitats for occurrence in the Pantanal and the Llanos ecoregions. When we realized or analyzed the environmental variables that were most influential on the models, they observed that the predictors related to mean temperatures have the largest effect. So as for the discussion, the combination of the statistical techniques associated with geoprocessing data has been used for some time in predictive models for ecology, especially in the ecological niche models in the context of macroecological analysis. So the models shown in figure 3 and 5 were designed to provide an analysis of the relative influence of the environmental predictors on the geographic distribution of melanism in big cats. So the main difference between the non-melanistic and melanistic models were restricted through the predictors or the moisture and temperature that was related to the thermoregulation in the natural habitats, suggesting that the presence or the frequency of melanism in the panthers may be regulated by the climate features. So in fact, this study has provided a characterization of spatial distribution of melanism in leopards and jaguars and demonstrated that such distribution is non-random. So the recent analysis have indicated that the melanism can reach very high frequencies in some leopard population, just like in Southeast Asia. In addition, there have been confirmed reports of melanistic leopards in India, Abyssinia, Ethiopian highlands, Java, and Malaysia, a Verder, Mount Kenya, highlands of Nepal, as well as a doubtful occurrence in the South Africa. This observation is restricted to some areas and may support the hypothesis that melanism can provide an adaptive advantage in certain ecological conditions. So, however, the exact geographic range of this coloration phenotype has never been mapped in leopards in general. So, the moist forests, especially in the Southeast Asia, presented very high frequencies of the melanistic leopards. So furthermore, there were no confirmed melanistic leopards in the Middle East, in the Arabian Peninsula, and in the central China. So overall, there was a significant reduction in the frequency of melanism in some biomes that consist of open habitats or temperate forests. There thus is a clear pattern in which melanism tends to increase in the tropical or subtropical moist forests and decrease in open, dry, or dry temperate habitats. The most part of melanistic jaguars was recorded in South America, especially in the Brazil, in Ecuador, and Peru territories. So the species potential distribution map and melanism distribution map indicate a high habitat sustainability in the moist areas. So as we know, the Amazon region has a large size and high population density of the jaguars and can be considered as a core habitat for the species. So although jaguars are often documented in the North and Central Americas, there were historically only 4 records of melanistic individuals in these regions. So as for the conclusion, given the current genetic evidence that indicating the population structures in leopards and jaguars have historically compromised a broadly connected population across its distributions, it would be expected to find a random distribution pattern of melanism throughout their ranges if this variant was selectively neutral. So the results of this study indicate that this is not the case and demonstrate that its distributional pattern is non-random. So although the observed punctual cases of increased frequency of melanism may be indeed derived from recent drift-induced shifts in locally isolated populations, they do not find support for a drift-based explanation for the observed pattern, and thus favor a scenario implying an effect of the natural selections under the polymorphic phenotype in big cats such as melanism. So, and at least there are some of the underlying differences that could be related to environmental predictors, possibly for the thermoregulation issues. In this context, it is relevant to note that the ecology of some species can drive genetic diversity, and phenotypes in natural population can present and spatial patterns of variations that was associated with the adaptation of or to the some environments that was regulated by the natural selection. So by connecting the coloration diversity to environmental information, which may have a direct or indirect influence on the adaptive significance of coat color variants, so it is possible to investigate the evolutionary processes that was involved in the generation and maintenance of polymorphic phenotypes in nature. Thank you.